Morning. Welcome all of you to attend our web training Tuesday. This session, as Miranda says, on how to reduce aflatoxin contamination using AF36. Again, my name is Guangwei Huang. I joined Almond board 2001. Since then, I oversee the food safety almond quality research at Almond board. We developed the mandatory pasteurization program and then expand our research to almond quality, understand how to use almond, how to preserve almond quality, what the uniqueness of almond. We expand our effort on biomass utilization. Food safety always our good focus, even though aflatoxin is not huge issue to almond, but there is some prevalence there. As we understand, AF36 is a very proven, very effective commercial tool used by other industry, developed by cotton industry, and then widely used by pistachio industry. So we have invested research, understand how this the AF36 will help the almond grower to managing those aflatoxin prevalence in the almond orchard. So today, we have fortunately, we invited 3 speakers who are intimately involved with this AF36 from invention and to application and evaluation of efficacy. So you will have a great chance to learn everything about the AF36 from today's session. So first speaker will be Dr. Themis Michailides, a researcher from UC Davis and the Kearney Agricultural Research Extension Center, followed by Dr. Bob Klein, and who oversees managing the California Pistachio Research Board. And then the session going to end with Dr. Leighton Liesner, who's the executive director of Arizona Cotton Research and Protection Council. So each of them going to have a certain content to cover. At the end of each presentation, if you have an urgent question, you can raise like 1 or 2 questions, and otherwise we're going to save. You can save those long urgent question to the end. We have at the end of our presentation, we have allocate the needable time for Q&A session. Okay, now I'm going to pass the flow to Dr. Michailides. You can share your screen.
Thank you, everyone, for being here to attend this session. My talk is on the ways to reduce aflatoxin contamination with AF36. And as you know, I need to say something before I present this presentation, that all the questions in the questionnaire are in the talk. So if you pay attention, you'll get 100% of the answers to the questions. Now, aflatoxin is the #1 toxin, mycotoxin, that is reported in the rapid alerts for various crops, including almonds. So, and also ochratoxins is the second in incidence. And you can see from this graph, the rapid alerts issued by the European Union range from 50 to 75, 76% for aflatoxin. So it's a serious problem, and the almond industry, along with the pistachio industry and the fig industry, have taken extensive measures and supported research pre-harvest and post-harvest to reduce aflatoxins and assure compliance with the aflatoxin regulations, and provide to the market a product with no free from aflatoxin or with very reduced levels of aflatoxin. The 2 fungi, what is aflatoxin? Aflatoxin is the major metabolite, a compound that is produced by 2 different fungi. And these are fungi, Aspergillus flavus and Aspergillus parasiticus. They produce these compounds several of these compounds. For instance, the Aspergillus flavus will produce the B1 and B2 aflatoxins, and the Aspergillus parasiticus, in addition to B1 and B2, will produce the G1 and G2. Among these aflatoxins, the aflatoxin B1 is the most potent. It can cause liver cancer and in some African countries, death actually of people and death of animals and things like that when they consume aflatoxin contaminated feed. So because this is the most potent toxin, the B1 is regulated by governments separately. And so this is an example of the tolerances imposed by USDA. For the total aflatoxins, 15 parts per billion. For B1 is 10. And the European Union for total aflatoxins 10. And aflatoxin B1, 8 parts per billion for pistachios and almonds and figs for direct consumption. Now, it is fortunate that this contamination is not very common event. It is very rare event. As you can see here, for almonds, 1 out of 25,000 almonds, or 35 to 35,000 almonds, will be contaminated with aflatoxin. So, and you can see in pistachios, 1 out of 5,000 of years, 1 out of 20,000 in on years, and figs, 1 out of 5,000. So it's a very rare event, and that is the difficulty we have in research to really reduce this rare event, even making more rare. So the life cycle of these fungi, Aspergillus flavus and parasiticus, live in the soil, and this is the life cycle of flavus, for instance, in an almond orchard. This fungi in the soil will colonize debris under the right conditions and produce conidia spores that become airborne. And also they produce these structures, these black structures that we call it sclerotia. And these structures are for survival of these fungi in the soil for a longer time. Now, this airborne conidia can reach the canopy, as you imagine. And now in the canopy, when we have damage by navel orangeworm, these nuts can be infected by the Aspergillus flavus and also parasiticus. Parasiticus will produce the same type of conidia, will reach the canopy. Now, what we know for the navel orangeworm is not only the larvae of the navel orangeworm will cause will produce the wounds for infection, but also we know the moths, they can carry propagules, and we trapped moths in almond orchards, and they're heavily contaminated with Aspergillus flavus and parasiticus. After harvest, some mummies will remain on the tree, and then during the winter, the mummies will fall on the ground, and we see some of these mummies actually infected with Aspergillus fungi, including the Aspergillus flavus and parasiticus, and that is how the way this life cycle closes. Now, when you do sanitation to remove mummies for reducing navel orangeworm, at the same time you remove mummies that are infected with Aspergillus, and in a way you reduce the inoculum to some degree. The Aspergillus flavus in the soil, as I said, produce sclerotia. And we have two types of strains, a strain that is producing a lot of sclerotia, very small sclerotia, and we call it the S strain, small, and the L strain that produces large sclerotia, L strain. So all the S strains are toxigenic, almost all. And in the L strain, we have an average of 1-to-1 ratio, toxigenic to atoxigenic. The Aspergillus parasiticus, all the strains of this fungus are toxigenic, producing aflatoxin. Now, in Arizona, there was a fungus, an atoxigenic strain, they call it AF36, that was selected there first, and it used on cotton and corn. And as we started doing research on atoxigenic strains to use them for nut crops, we found out that the one of the atoxigenic strains was very similar to the Arizona strain, and this was the AF36 in the same group of vegetative group, as we call it, And this was the most commonly found in almond and pistachio and fig orchards. While the other toxigenic strains were less than 1%, this strain ranged from 4% up to 7, 8%. So it was a good choice then to focus our research on this strain. And this strain, the toxigenic strain, which means does not produce aflatoxins, never produce aflatoxins, the way it works is when you apply the AF36 in the soil of the orchards, this AF36 will multiply, will produce spores and increase the population of the atoxigenic strain. Now, the atoxigenic strains then strain will displace or compete with the existing toxigenic strain population in the orchards. And that is the principle how this strain works. Let's take an almond orchard in this case, and where we have a ratio of about 1-to-1 atoxigenic and toxigenic native strains of Aspergillus flavus, and also we have Aspergillus parasiticus. Now, when these strains release their spores from when the spores reach the canopy, we'll have a high risk for some of the spores, the red spores here, the toxigenic, to produce aflatoxin and contaminate the nuts. When in an orchard, an almond orchard, we apply the atoxigenic strain, in addition to the native toxigenic and atoxigenic, now we added the AF36, so we increased the numbers of the green spores, the good spores, which do not produce aflatoxin, and the chance for the red spores to produce aflatoxin is reduced. That's the way it works. Now, but in order to get this material registered, we did a lot of research, almost 10 years plus research with pistachios primarily. We reached to a point where our research was approved for experimental use permit in pistachios, and we treated 3,000 acres of pistachios, and 3,000 were used as controls. At this point, I would like to thank the Paramount company, then and now Wonderful Orchards, that supported this research with sites and the library samples you will see in a moment, and still is continuing. The Wonderful Orchards supporting us with the sites of treatments and the area-wide project. So we, after treatment, what do we do with these applications? The F36, we found out that it became the dominant strain in the soil where it was applied. And you can see in 2008, the first year of the experimental use permit, treating 3,000 acres. After the application, we increased the F36, the toxigenic F36 in the soil about 45%. With the second application in 2009, we reached 70%. With the third application, by the September of that year, it increased up to 90%. In 2010, 95%, and 2011, we reached levels of 90 to 95 displacement, and this is what we need in order to have a good efficacy in reducing aflatoxin. All this analysis of the soil samples are done with the VCG assay, we call it. You will see a slide on this to explain a little more. It is very tedious, very time-consuming, and it is, and that is the reason why some of the results are delayed to report to your industries. Now, what happens then with we saw that the F36 became the dominant strain in the soil, but let's see what happens with the toxigenic strains. The toxigenic strains, the Aspergillus flavus, the S strain, and Aspergillus parasiticus here. In this, before application, about 65% were toxigenic in this, as an average in these orchards. And we see after application, we have some reduction here. And in 2009, some about the same levels here. This is the variation we see. But by the application in 2009, After this, the levels decreased very significantly down to below 10%. So this is a major reduction in the toxigenic population, toxigenic population of the pistachio orchards. Of course, we want to see the efficacy. We wanted to see the effect on the aflatoxin, and we analyzed library samples for aflatoxin. And the library samples are taken at the processing plant as the nuts are being unloaded. And after the processor finishes with the use of these library samples, we analyze them in the laboratory. And we found out that during the experimental use permit, as an average, by this analysis, we show a 40% reduction in the positive aflatoxin positive samples. You can see it here, very significant reduction, but not up to 80, 90% that they get when they treat cotton or corn or other crops, row crops. So in 2012, this product, the F36, was registered. And after registration, we continue monitoring the same experiments, the same fields. And we can see here in 2013-15, we had very high levels of reduction, more than 60 to almost 80%. But in the last years, we see a reduction in efficacy of reducing the efficacy below 40% that we achieved with the experimental use permit. And now we try to explain the various reasons. And probably you see in a moment, one of the reasons is that the sporulation of the product is not as good as it used to be during those years. So the product was registered as F36 initially in 2012. 10 pounds per acre is the rate of application, and the application timing is late May to mid-July. Here is a supplier and applicator, Western Milling, and another applicator here, Agri Systems Incorporated in Chowchilla. Now, let's go. Now, in 2007, the almond industry approached us, and they wanted to do more research on aflatoxin in order to include the almonds in the label, because the initial registration of the F36 was only in pistachios. So we started experiments in microplot experiments in this nonpareil almond up in Colusa County. And we did the same procedure, we collect the soils, we analyze them, the soils before, we collected the soils before application and after application, and then we identify the isolates in the laboratory using the VCG assay. Now, the VCG assay, as I said, is a very complicated assay. We start with the soil sample, we have all this, we make a suspension, initially, we use Petri plates, or we put soil, half a gram of soil per plate. And then we have 20 plates, and we get 20 isolates from each sample, 20 isolates of Aspergillus flavi group, in order to get significant numbers per sample and do statistics. So then we follow this. Once we get the isolates, we have to develop mutants. This is a little more complicated. These mutants, and pair the mutants with the unknown strain here. And when we have these barrages here with the two mutants of Aspergillus flavus, then this indicates that this is an F36. So it is a complicated method. It takes a long time, up to 2 months. Recently, though, we developed this PCR analysis method, which takes only 7 to 10 days. And in 2020, we use some of the samples with this method. And in 2021, all the samples will be analyzed with this method, which is very quick. And it has the advantage also, this method, not only to show us the percent of F36, which is these bands below this line, but also percent toxigenics, which is the bands above this line. So you can see that the procedure is improved now and is faster. But going back to the orchard, the almond orchard, we did the same thing. We collected samples, and we noticed that the AF36 again became the dominant strain in the soil of the almond orchard, very similar to what we had in pistachios. After application here, we start from a level 5% of the F36 natural levels, and now we have about 95% in September of 2007. This level maintained until July of the following year, and with the second application, we have a little increase close to 100%. And then 1 year we did not apply it, and the level maintained for a second year very high, and then start going down. Once it went down, we apply another application in 2011, 2012, and you can see the levels of toxigenic F36 are increasing. And so there is also an effect in the untreated control because these are microplots, as you can see. But you can see that the F36 becomes dominant in the orchard. We also monitor what happens with the toxigenic. This orchard had very high levels of parasiticus. So we had up to 90% toxigenic strains in the samples. And you can see this is before the first application of F36. And the population went down below 10% here with the first application, the second application even lower, below 5%. And then once we saw an increase in the toxigenics, we applied another application in 2011. The level is still high here, but after the second application, another application in 2012, we see the decrease going down. So the F36 reduced the toxigenic strains in the almond orchard, very similar to what happened with the pistachio orchard. The toxigenic Aspergillus flavus limits the amount of the high aflatoxin producers that become associated with the crop. We also did efficacy studies in the laboratory using co-inoculations. We inoculated almond kernels with the toxigenic alone Aspergillus flavus and with AF36, and the toxigenic Aspergillus parasiticus alone, and also the parasiticus with the F36. The results were very impressive. When the Aspergillus flavus alone was inoculated on the kernels, we had very high levels of aflatoxin. The same with Aspergillus parasiticus, much higher. But when these strains were co-inoculated with the F36, you can see the levels have been reduced. Also, because the Aspergillus parasiticus produces G aflatoxins, it produced in these samples G1. You can see alone a high level of G1, but with F36 and very, very small levels. So the efficacy was good, very good in co-inoculation experiments, and all these data were used for the registration of almonds and also the figs by the EPA and DPR. So the conclusion from the AF36 studies in almonds, pistachios, and figs can be generalized like this. The AF36 is widespread in California nut crops and fig orchards. It becomes the dominant strain when it is applied, persists well in the soil, particularly in the almonds, persisted for a second year. And now with if the growers are applied in the field, we'll also determine if it is necessary to apply it every year or every other year. At least the initial indications was that it persisted very well for a second year. It did not increase crop decay. This is very important because if you don't want to apply something that really will increase the decay of the nuts, the infection of the nuts by the fungus. And we had similar results for the 3 crops. And the F36 was extremely effective in reducing the aflatoxins in the co-inoculation experiments I showed for almonds, but the same results were for pistachios and figs. All these results we got were helpful for the registration of their product, Aspergillus flavus AF36, but now has the name AF36 Prevail. because although the initial product was the carrier of the initial product was wheat, now it is sorghum. And the registration was done on the 7th, the approval was on the 7th of August, 2017, not really given the opportunity for treating that year. But the next year, it was available for the almond growers to use. After the registration, we were able now to do some experiments in commercial orchards to determine the best timing. And we did an experiment here where we applied in from June 26, July 10, July 24, August 7, and then we monitor the sporulation. You can see these dates when the product was applied on July 10 and July 24, we had the highest levels of sporulation. And also for these dates, July 10 and July 14, we see that we had the highest levels of AF36 reaching the kernels of the almonds. So there is a good correlation of the amounts of sporulation we see on AF36 in the soil and the amounts of AF36, the propagule of AF36 that reach the kernels. It's a very good linear correlation. We also showed that these applications reduce the toxigenic fungi that reach the kernels of almonds. And in fact, in this case, this is the untreated control. In this case, the July 10th was the best application because it reduced the toxigenic on the kernels the most. Of course, after the registration of the products, the pistachio industry started with 75,000 treated the first year, 150 the second year, 2013, and then we have 200,000 acres treated every year since then. But the almond industry did not adapt these applications so well. So in 2019, the Food and Quality Committee of the Almond Board of California approached us, and they wanted to do a major study in 5 regions, McFarland, Woodland, and also in Williams, Madica, and Turlock by treating 100 acres and having also 100 acres untreated in each location. And this was to show that this product will have an effect in reducing aflatoxin in almonds. Again, in 2019 and 20, we used this tedious and time-consuming method. But as I said, late in 2020, we started using this. It works really well. In 2021, the project has been also approved. We'll use this method and we'll have the results available to the industry. But let's see what happened with the first year of application in 2019. Those 100 acres of treated fields in 5 locations. Yeah, this is the general, all the results from that year. In 3 locations, McFarland, Manteca, and Woodland, you can see this is the control, and this is the treated fields. You can see a reduction of the toxigenic. The red bars are the toxigenic. The green bars here are the L strain. That L strain has also toxigenic in there. But you can see we increase this bar here and decrease the red bar. In the Manteca, the same type of result, major reduction here. In Woodland, again, even more, a reduction of the toxigenic strains. Now, if we take and analyze determine the strains that we recover from these fields and compare the L strain alone now here, the L strain before treatment here, and in 2020, okay? This is 2020. This is the control, the untreated, and this is the field that will be treated. And what happens is these are the levels of toxigenic here. And you can see here after the application of AF36, after treatment, you can see there is a reduction from the control up here to down here in 2020. Now, if you take the L strain and you wanted to see what is going on in here, how much toxigenic, toxigenic were in these composite bars, and you'll see now that before treatment, these are the levels of toxigenic in that L bar that has also toxigenic and toxigenic. And these are the levels of toxigenic before treatment. After treatment, you can see this is the untreated control, and this is the treated field. This is for all 5 locations. You can see here we have a reduction about 40% from the toxigenic to the atoxigenic within the L strain. So, and also, if you take now all the toxigenic strains of Aspergillus flavus, the S, and Aspergillus parasiticus, also the toxigenic strains L of Aspergillus flavus, here's the situation here. Before treatment, you have very high levels of toxigenic. These are the toxigenic. This is the total, okay? The F36 is the toxigenic here, but after treatment, you can see a major reduction of the toxigenic and a major increase of the atoxigenic populations. So this reduction here is about 40%. Remember, in the reduction of the pistachio aflatoxin positive, that was 40%. I wonder if this is a coincidence or it is a true event. 40% reduction in the toxigenic, 40% of the aflatoxin positive pistachio samples. We don't have results for almonds. But we analyzed really 228 samples of almonds, but the aflatoxin was very, very sporadic. Only 10 samples were positive. The numbers in parentheses are shown in the samples either negative or positive for aflatoxin, very, very few. The range was from 18 to 64 parts per billion. But in one field, these are analyses, not only samples from the field, but also samples from the processing plants, and also samples from rejections. We saw a difference here only in the rejected nuts, the nuts that were, the rejected nuts that came from fields that were treated had 1 positive, and the rejected nuts that came from fields that were not treated had 4 positives. So the aflatoxin almond project in the 5 regions showed that the application of AF36 Prevail displays greatly the toxigenic strains of Aspergillus flavus and parasiticus in commercial orchards now. So this is a good incentive for the growers to actually follow and apply this material. The aflatoxin positive samples were too few to make any conclusions, but in one case we had in rejected nuts that came from a field that was treated were fewer, only 1, than those from the fields that were not treated. The plans here and the committee, the Food Safety and Quality Committee, is to expand the project. They approved this project to increase the treated acreage to 5,000 acres. And this we will get more soil samples to show again the displacement. And most importantly, we will we'll take samples, twice as many samples of nuts for analysis for aflatoxin. And of course, this is a major experiment. We need to increase the personnel working in this project. And with that, we'll give more information to the growers, educate the growers, and on the benefits of treating their almond fields. So the story has as follows. Okay, aflatoxin contamination risk in nuts and figs depends on 3 major parameters. The host. We have the almond, we have pistachio and figs. They're all susceptible to the aflatoxin. We cannot change them at this moment. What we can change here is a pest that really is instrumental for aflatoxin contamination, the navel orangeworm. We can reduce it by controlling the navel orange worm. The environment, we cannot change the environment. We need irrigation, the moisture that is needed for sporulation of these fungi. We cannot change this. What we can change is the causal, the pathogen that produces aflatoxins, bring here the AF36, the toxigenic AF36, and remove the toxigenic strains. And that way we reduce the risk for aflatoxin contamination in nuts. Fortunately, also there is another product, the Aflagard, that is produced by Syngenta. It is an etoxigenic strain, different etoxigenic strain, NRRL 21882. And according to Syngenta, the registration in California for use in pistachio and almonds is expected soon. It is now in the 30-day comment period, so it has been announced by CDPR. The difference, the Aflagard, the carrier is barley. Remember, the AF36 Prevail is sorghum. And we did some studies to support this registration. First experiments we did were to determine how different are in the sporulation of the Aflagard, Aflagard in one side and AF36 in the other side. And this was done under different temperatures and different soil moistures. 21% is the saturation moisture of that soil that we use. And you can see the optimum sporulation for both occurs at the saturation point. And we checked two temperatures here, 95 Fahrenheit and 86 Fahrenheit. And in general, the solid lines are the Aflagard and the dotted lines are the F36. The sporulation of F36 is lower than the Aflagard. Also, we noticed that under drier conditions, as the moisture decreases, again, the Aflagard maintains higher sporulation than the AF36. And you can see also that the optimum sporulation temperature was at 95 for both, but still the Aflagard sporulate much faster, better, higher levels. And here you can see that the Aflagard sporulates faster also. Under this night temperature 59 and optimal humidity, after 4 days, the Aflagard was covered with a sporulation. The AF36 Prevail is just starting. After 1 week, you can see a little less sporulation in the AF36, while the Aflagard is loaded with sporulation. Here is under 100% relative humidity. You can see the Aflagard is really, these are 2 seeds per well here, under 100% humidity, 2 seeds of Aflagard, 2 seeds of AF36. You can see a big difference in sporulation. And this is because this is a material that is fermented, inoculated with a fungus. This material here, sorghum is the carrier, which is coated with the Aspergillus flavus F36, which means it is perhaps that makes some difference. So we do have several tools for reducing aflatoxin contamination of almonds. The first tool is sanitation to reduce mummies and also apply insecticides to reduce to control navel orange worm. The second is the application of AF36 Prevail, which the product is registered. It's ready for the almond growers to apply it, especially after our commercial, the large experiment with 500 acres of treated almonds. We know the optimum time for the best sporulation. It's mid-July. The label says late May to mid-July, of course, to account for large farms that need several days to cover the entire acreage. Also, post-harvest, proper stockpiling to avoid moisture and leaks under the plastic cover. It's important. I did not cover this due to limited time, but we did some studies before. And the fifth tool on its way, it's the Aflagard that hopefully will be registered by July this year, that is produced by Syngenta and shows better sporulation under these orchard conditions in California, better sporulation qualities, like sporulates better even in drier soil and even in lower temperatures. And with this, I would like to thank several people here who work in our lab, and also Guangwei and team and Ashley, who helped us initiating the project in 2019, and a lot of the growers that and managers that participated in this 5-region experiments, major experiments.
I may speak with you today. My presentation is very, very short, not nearly as technical as Themis's, and probably not going to be as technical as Leighton's either. So I should be putting us back on schedule pretty well. As has been commented, the pistachio industry has been using the AF36 since about 2011, 2012. We do have sizable coverage on our acreage, and the way we go about getting growers to use it is a little bit different than other industries. But I wanted to go back a little bit just to cover, for everybody's benefit, what the history of AF36 is and this whole approach of competitive exclusion. And back in the early 1990s, the USDA formed the Aflatoxin Elimination Working Group, and they funded it with $1 million. And the commodities that were present in the working group included tree nuts, cotton, peanuts, and corn, the commodities that had aflatoxin contamination issues. Among the tree nuts, we had almonds, pistachios, and walnuts. It was quite an unusual project in that industry largely decided what proposals were going to be worked on. We had a proposal period, and then we reviewed the proposals, but each of the different commodities then decided which approach they wanted to follow. And corn did mostly a breeding program. Peanuts did a breeding program, and they worked on what ultimately became Aflagard. Cotton worked primarily on AF36. Walnuts and almonds approached it from breeding, and pistachios looked more at Aspergillus biology. So most of the a sizable amount of the work, I can't say most, but a sizable amount of the work that Themis reported on Aspergillus biology was funded out of the Elimination Working Group, and then out of also the California Pistachio Commission. So out of this program, you can see what we've gotten is AF36 and Aflagard. The breeding efforts, and as any plant pathologist would tell you, and I'm sure Themis would agree with me, the breeding programs were a complete waste of time, at least as far as aflatoxin has to go. But cotton and Texas Gulf corn worked primarily on the AF36, and the AF36, as mentioned, was produced by the Arizona Cotton Control and Research. Got the label all wrong. And then pistachios were added to the label in 2011. So pistachio industry is quite different than almonds because we have relatively few handlers. At the time, it had 7 handlers that accounted for 95% of the industry. We now have about 9 handlers that account for 95%. So we're seeing some slight changes, but largely we have, we talk to everybody basically in one room when we have a meeting. And aflatoxin was a significant trade issue for all the handlers, particularly because of the EU and the history of aflatoxin in the EU. And so the handlers can see direct benefit from the reduction of aflatoxin, because they don't have to deal with it then in their export markets. And the growers see some indirect benefit, primarily because we have better market access, and their crops can maintain a higher price. The other advantage with AF36 is it's inexpensive, and it's very quick and easy to apply. If you want to see how it was applied when we first did it in 2011, 2012, you can go to the California Pistachio Research Board website, calpistachioresearch.org. And there's a training video that shows how it was applied when it was on wheat seed. Done off the back of a quad, and the quad can fly along at 10 miles an hour without any trouble through the orchard. And so a single quad can cover a lot of acres in a day. The AF36 was inexpensive. It was $1 a pound and 10 pounds per acre applied on the berm. So it was quite easy to do. The handlers, remember those 7 handlers accounting for 95% of the industry, subsidize the application of AF36. And they do it by different methods, depending on which handler you're talking about. Some of them buy the product and then pay for the application. I think probably more common is a price premium, where a premium is added to what the grower receives for their product. In general, the price premium that they receive on their product accounts for about twice the cost of the material and the application. So growers are actually being paid to apply the material. And then sometimes there is through a direct reimbursement system. So Themis was discussing there about efficacy. And so obviously the question is, does it work? And right now we have a question of how do you measure success when virtually the entire industry, the entire pistachio industry is applying it to all their acres. We can look at the displacement in the orchard, and Themis there noted in some of his slides that we have in many cases, we have 95% displacement of the toxigenic strains within an orchard, but we're still only seeing 50% reduction or so of aflatoxin in the crop. So why do we get 95% displacement with only 50% control? My answer to that is that the fungal spores that the crop sees in the orchard is not just derived from what is present in the orchard and in the soil underneath. There's a lot of stuff that gets blown around in the valley. And, you know, for pistachios, which of course harvest later than almonds for the most part, when you guys harvest almonds and you blow up a lot of dust, that's just not dust, that's a lot of fungal spores too. So that stuff blows into pistachio orchards or into other almond orchards for that matter, and spreads toxigenic spores from non-treated areas into treated orchards. So I think AF36 will work much better if you look at it on an area-wide project where all the almonds and all the pistachios in a given area are treated to reduce that influx of toxigenic spores from non-treated areas. So we still say, does it work? And it's still supported by our handlers, because aflatoxin is a significant concern. The cost of application, well, the total cost across all the acres of pistachios is significant. The cost of rejections in the EU is also costly, but also it creates very good public relations with the regulators and the trade in the EU. When you can talk about biocontrol of a toxin, that is a strong positive talking point. Themis also discussed matching the environmental conditions to sporulation with Aflagard versus AF36. We'll have to see how that all works out as we go forward. So my brief take-home message is that AF36 is a very useful tool to manage aflatoxin, but it is not a panacea. As Themis mentioned, navel orangeworm damage is a significant driver of aflatoxin, and consequently, we have to look at navel orangeworm control to effectively manage aflatoxin as well. But it is an important adjunct that can serve to reduce some of those major spikes that we see with fluctuating navel orangeworm populations. And that's all I have to say. So I'm open.
Thank you for having me. It's great to be here. Haven't had much contact with the industries during this COVID period, but hopefully we're coming out of that and we'll be able to do more outreach and more talks with you guys, or provide more information as it's available. Themis already covered a little bit of this. What are mycotoxins? First identified in the 1960s, and they are a metabolic byproduct of fungal growth. So these fungi that are living in your field, getting in your crop, just a part of their natural living is where these toxins come from. Of course, different fungi produce different toxins, as Themis said. And still today, function of that toxin for the fungi is poorly understood. Can be concentrated through processing, highly toxic to humans and animals, most commonly linked with liver cancer in humans, and of course the impact it has on our ag producers is reduction in quality and loss of revenue. So this is our slide of, on the left-hand side, my left-hand side, S strain A flavus. On the right-hand side, an L strain or typical A flavus. And of the L strains, there are some that make a little bit of toxin and some that make no toxin. Generally, all the S strains make a lot of toxin. AF36 is part of the L strain group and is incapable of making toxin, and was the 36th isolate that was looked at by Dr. Peter Cotty, who invented this technology that we worked with to develop this product. So something I like to show growers is this series of 3 slides. Think of the square representing a part of the topsoil in your field. And excuse me, the different dots represent the different fungi that inhabit those soils. So generally in native soils, we find higher levels of toxin producing strains. We also find atoxigenic AF36 at low levels, and then of course other fungi. And I also want to note that AF36 is native to every soil in the US we've looked at. And so it, in my opinion, is better in tune with the environment. To our knowledge, the fungi used in our competitor's product is not native in California soils. But as you see, this is an untreated soil. Treat 1 year and begin to see a change. We're changing the propensity of atoxigenic to, or toxigenic strains to atoxigenic strains. And with repeated treatments, you get a buildup after annual treatments. And what we find is if you stop treating, regardless of which technology you use, the system is going to want to turn back to equilibrium. And the atoxigenic, without extra help, will be reduced over time back to the natural state of the soil before you started treating. So what is AF36 Prevail? As was mentioned, it's a carrier, sterilized, devitalized milo, carries no other pathogens, no human coliform bacteria, coated with an atoxigenic strain of the common fungus Aspergillus flavus, common in most soils. Again, it was the 36 isolate that was looked at by Dr. Cotty when we developed this product. And it is an effective biological control agent for the displacement of aflatoxin producing fungi. As mentioned before, it works by displacing or filling the niche occupied by toxin-producing strains, S strains, thereby lowering toxin levels on the crop. AF36 does not increase the total fungal population, which is an important point. When the carrier, wheat or sorghum, we can use either, comes in contact with moisture, the AF36 begins to grow and produce spores to be dispersed by wind and insects, is how these spores move. We initially used wheat in a different process. And one of the things that I haven't heard mentioned yet today is one of the reasons we use sorghum seed is when you look at a wheat seed or a barley seed, milo seeds are about a little over two milo seeds plus a little bit to one wheat seed or barley seed. The reason why this is important is what we found is more point sources in the field are more important. So that is why our product is on milo instead of wheat seed these days. So how do you really want to measure the effectiveness of AF36? Well, aflatoxin levels alone are what everybody's interested in, but they're an incomplete measure of effectiveness. Testing soil levels of L and S strains before application, along with soil and crop samples after harvest, will measure displacement. And why is this important? In high aflatoxin years, displacement can result in dramatically reduced aflatoxin levels, though in some instances, those will still be above the optimal levels for your market. And this is something we've seen with the technology from day one. It doesn't matter which technology it is. In bad years, if you get a 90% reduction in aflatoxin and you would have had 1,000 parts per billion, you're still over any levels that you want to see. And of course, we use VCG, vegetative compatibility group analysis, as Themis was mentioning, to measure displacement of aflatoxin producing fungi. We're also working on a PCR or have been using a PCR process to speed up samples. We're still going to be pursuing refining that, as we find that for now, the VCG is still more accurate than the PCR is. And this is just another diagram similar to what Themis showed of the VCG process. It's a long, involved process, and it is much more expensive and costly in time and manpower than the PCR. So hopefully we'll get to a point where PCR will be the gold standard, and we can get the accuracy up where we're feeling more comfortable with it. So one of the other things that I haven't heard mentioned today is that through the research that we've done and ARS has done, our partners have done over the years, what we think we know is that the action of disturbing the soil for ag purposes, for growing crops itself, somehow encourages the toxin producers. The reason why that is not fully understood, but it's something we've seen again and again. And so unfortunately, we're in the process of doing what we need to do as ag producers. We're also creating some of our own problem. And that's why we work on projects such as these. Excuse me, allergies. So, and I think some of Themis' slide alluded to this too, and I apologize, this is cotton. I know we're talking about tree nuts, but it just reinforces the point that as the crop biomass increases, so do the conditions for aflatoxin contamination. So the time when these fungi are most active is during the warmer times of year when the biomass is increasing greatly in the field. So one of the challenges I've always had when talking to growers or PCAs or other people is getting across the idea of how these fungi are dispersed in the field. So I'm going to try to show a short video here. I tried to do this yesterday and it was a little bit interesting. I'm going to have to change my sharing.
Actually, Leighton, we can hear the sound. Oh, you're good to go.
Yeah, but you can't. But you couldn't see the video, right?
Oh, we could see it. Mm-hmm.
Oh, you could.
Mm-hmm.
Okay. So this is the micro level that you're looking at, where the fungus is growing, which are being knocked off and dispersed into the environment. So I hope that came through for everybody. Are we still online?
Yes.
Yes.
Okay. So moving forward, you can see this is just an example of the two different ways that we can coat onto wheat or milo. These are both AF36 Prevail, just on different substrates. These are what spores look like retrieved from a pistachio orchard in Arizona. So great sporulation there in the field. So a little more on history. The council is a not-for-profit state agency of Arizona. And when this technology was developed, there was some decisions that had to be made about how we wanted, what the growers wanted to do with it. And it was decided so that this technology would not get bought and shelved or priced at a level that would cause barriers to use. We would produce and sell the product ourselves. And fortunately, we have the special ability to do that because of our statutes. So first pre-registration meetings in 1993. Of course, fast forward to 2017, Prevail approved for almonds and figs. We're currently working on a organic version, and we're also working on different application methods to be used in the different crops, particularly in tree nuts, which I hope we'll have in 1 to 3 years. So what are the goals of our aflatoxin mitigation program? Of course, expand atoxigenic strain technology, improve and simplify manufacturing, which we continue to do, add new strains, and effectively deliver those either in singly or in mixtures. And that's what we think what may be the future is mixtures of strains. Ensure the long-term availability of atoxigenic technology and provide atoxigenic technology to the commodities that need it economically. And that is how our reach has grown, is through partnerships, first with corn and then with tree nuts. And now we're moving into other states, expanding across the U.S. This is a sample of our label, just a simple label, no flashy advertisement. And our catchphrase that was coined years ago was "For growers, by growers." Another thing that we do is research, and it's probably our primary goal. And this is a picture of our evaluation lab. We do soil seed and have done air samples in the past in this lab. And this is where generally all of our data is generated, along with some of the work we do cooperatively with Agricultural Research Service. So this is some data from Bowie, Arizona, and pistachios over 3 years. And what we saw using BCG analysis is an increase of 380% AF36 over that period when 4 pistachio orchards. So from 15% natively to 72% after treatments. And as Bob was saying, one of the important points that is often missed is the area-wide effect of atoxigenic technology. And if you look on the left-hand side of the screen, you can see that prior to treatment, the treated field had 1% AF36, varying levels in fields surrounding it. After treatment, the treated field was at 92% AF36, and the other fields had increased dramatically, even though they were not treated. So if it's the atoxigenic or the S strain, whatever's dominant is not only affecting the field that you're looking at, but has a wide effect over a large area, as Bob was saying. So it's one of the important reasons why the area-wide effect of everyone treating has such a better outcome for all involved. So this is a Google map of the growing area in California, and the yellow pins that you see there represent sites where we've been sampling since 2018 to look at issues surrounding AF36 and efficacy and the 2 different crops as well. And we compiled that data late last year and produced a report, which is available on our website. And this is one of the slides from that report. And what we see is in pistachios, amongst those sites, we have an average 67% AF36 above the native levels. S strains and A. parasiticus are greatly reduced over the original levels. And we don't see the same kind of performance in almonds, of course, because almonds hasn't been treating at the level that pistachios has. But even at that, we have seen some effects in almond orchards that are close by to pistachio orchards that have been treated. So again, the area-wide effect showing itself. Another thing we've done is leaf samples in these orchards. And our latest numbers show that we were able to recover 30% in the leaf canopy, not just in the soil, but in the leaf canopy. And of course, almonds hasn't been treating, because they don't have as high of a presence of AF36. So this is over time. The first year we did just soil sampling. And then in 19 and 20, we did leaf sampling and soil sampling. Large numbers of samples, you can see at the bottom of the slide. And again, what it shows is we have high sustained levels in pistachios and almonds lagging behind, probably because it hasn't been fully adopted in that crop yet. So what's the take-home message? Prior to treatment with AF36 in tree nuts, the natural occurrence of AF36 in pistachio orchards across the region was between 2.9 to 15%, with an average of 7.4, Doster et al. 2014. Now occurrence of AF36 is well over 60%. So how do we maximize efficacy of AF36? First, we have to realize that it is a biocontrol, it is a living organism. Have to have available water, protection from sunlight, and sufficient canopy, because UV is deadly to fungus. Proper timing of application and a conducive microenvironment are key. Product support, as far as the Cotton Research Protection Council, we do pre-testing soil sampling that gives us a baseline on what your levels are of toxin producers versus atoxigenics versus other fungi. And then we also do post-harvest testing to determine what the displacement in your field is, showing how well the product has changed the makeup of the fungal environment in your field. And of course, we can make individual recommendations for local climate and growing conditions. When to apply, and I apologize, there's cotton and corn on here, but we do service several commodities. Pistachios, almonds, and figs, late May through early July, as others have said. Of course, when sufficient moisture is available or will be available within 3 days of application, and after any cultivation is complete so that you're not covering the product up. These are some of the methods that we've played with for dispersing AF36 into the fields. Of course, you can see the herd spreader on a four-wheeler, which Bob talked about. The rest of these are test rigs that we've experimented with. This machine belongs to a grower in Bowie, Arizona, Dr. Cook. It's a really neat machine. He's able to treat 2 rows at once and adjust the speed of those belts so it throws the product right into the wet zone. So potential for future research, continue to improve the commercial production technology, which we're doing. In fact, we're in the process of building a new facility with capability up to 25 million pounds a year. Improvements in application technology. Additional toxigenic strains be used as mixtures for unique geographical application. Label expansion for additional crops. And expansion of use in additional states. Of course, the emphasis is on low cost and high quality and maintain the for growers by growers philosophy. So, and the other comment that I'd like to make, and this is not anything against Themis, but when you look at sporulation, and particularly at 59 degrees Fahrenheit, to our knowledge, that's not the time in the field when it's most important to have sporulation. Your average temperature is going to be much higher at those times. And what our research has shown again and again over time is that as humans, we tend to think that more is better, but more sporulation does not equal more displacement in the cases that we've looked at. So, but I will say that what we do know is that different strains being used in the same area, in other words, mixtures of strains, tend to have a better overall outcome for growers than just single strains. So this technology is the only technology we really have for addressing aflatoxin in the field. The council is happy to be able to support agriculture on a broader scale than just cotton. And I appreciate you guys allowing me time to talk to you today, and I hope I didn't take too long. I apologize if I did. Thank you.